Welcome to today's Explainer. We're jumping right into the thick of it today, rigorously analyzing the molecular evolution, the pathological tropism, and the current countermeasures against a virus that is frankly rewriting the rules, highly pathogenic avian influenza A, specifically H5N1 Clade 2.3444b. This thing has aggressively pushed its way out of avian reservoirs and straight into mammalian populations, creating a truly unprecedented global panzootic. So we're going to get highly technical today. We'll explore the exact shape of its receptor binding pockets, the brutal clinical realities of acute respiratory distress syndrome, and the cutting-edge mRNA 1018H5 vaccines currently in clinical trials. Let's get into it.
Here is a quick look at our agenda. We'll start with molecular virology and evolution, move right into receptor tropism, map out pathogenesis and clinical outcomes, track the zoonotic transmission routes, and finally wrap up with medical countermeasures and vaccine development.
All right, Section 1, Molecular Virology and Evolution. Let's look at the genetics of H5N1.
To really understand the threat, you kind of have to look at the timeline. It perfectly highlights this stark, rapid shift from an avian problem to a full-blown mammalian panzootic. Back in 1996, we saw the ancestral goose/Guangdong lineage pop up in China, and then bam, 1997, the first human spillover in Hong Kong. But the real structural game changer, that hit in 2013 with the evolution into these highly diverse Clade 2.3.4.4 variants. Fast forward to early 2024, and we've got the first multi-state spillover into U.S. dairy cattle. Which brings us to today, 2025 and 2026, where we are dealing with the absolute dominance of Clade 2.3.4.4b genotypes. I'm talking specifically about variants like B3.13, D1.1, and D1.3.
Now, what makes this specific clade so incredibly fit? Well, its evolutionary divergence is exactly what allows it to infect such a massive spectrum of wild and domestic species globally. Through non-stop genomic reassortment with low pathogenic wild bird flu viruses, Clade 2.3.4.4b has essentially hacked its own fitness. It's constantly swapping genetic material, like shuffling a deck of cards, to optimize its survival and replication across totally different environments and host species.
Moving right along to Section 2, Receptor Tropism and Specificity. Let's talk about binding specificity.
Okay, so here is the absolutely crucial molecular barrier holding this virus back. Current H5N1 strains bind strictly to avian-type alpha-2,3 sialocytes. And thank goodness, because that's what prevents efficient human-to-human transmission. You see, these avian-type alpha-2,3 linked sialic acids are abundant in birds, but in us humans, they're mostly hidden away deep in our lower respiratory tract and on our conjunctiva, the mucous membrane of the eye. Conversely, for a flu virus to spread easily between humans, like through a sneeze or a cough, it absolutely needs to bind to human-type alpha-2,6-linked sialic acids. Those are the ones heavily coating our upper respiratory tract. Until the virus adapts to those alpha-2,6 receptors, widespread human transmission is going to stay highly inefficient.
But we have to pay close attention to the data. Looking at recent glycan microarray data from a severe human infection in British Columbia involving a genotype D1.1 variant, we see proof that these recent variants are actively broadening their avian receptor spectrum. They're now accommodating new structural shapes like alpha-1,3 fucosides and bisecting glycine C. But, and this is a massive but, they still show absolutely zero adaptation to human alpha-2,6 receptors. If you compare the BC subpopulations to that older 2004 Vietnam control strain at the bottom, you can clearly see the virus exploring a much wider array of avian glycans. It's highly adaptable, sure, but it hasn't crossed that human receptor threshold just yet.
To track that threshold, we have to talk about the HA190 delehelix. This is a critical structural element in the hemagglutinin and glycoproteins binding pocket, and mutations right here are exactly what global surveillance teams are sweating over. When we see amino acid substitutions in this specific pocket, like the E190D mutation detected in recent clinical samples, it raises massive red flags. Switching receptor binding specificity via these precise molecular changes was a huge factor in past pandemics, so continuously sequencing this specific helix is literal frontline defense.
That brings us to Section 3, Pathogeneses and Clinical Outcomes. Let's face the clinical realities.
When spillover does happen, the pathogenesis is stark. We frequently see it escalate incredibly fast, going straight from localized ocular symptoms to acute respiratory distress syndrome, or ARDS, and systemic hypercytokinemia. Now you might be wondering, why the eyes? Well, remember those alpha-2/3 receptors we just talked about? They're right there in the conjunctiva. That's exactly why we've seen so many dairy workers presenting with isolated conjunctivitis after getting splashed with contaminated raw milk. But if the virus makes it down into the lower respiratory tract, where we also have those receptors, it can trigger severe, community-acquired pneumonia that just laughs at standard antimicrobials, eventually spiraling into a totally life-threatening cytokine storm.
Actually, there's a foundational paper from Nature Medicine back in 2006 by De Jong and colleagues that hit the nail on the head. They noted, fatal outcome of human influenza A, H5N1, is associated with high viral load and hypercytokinesia. This perfectly highlights that H5N1 mortality isn't just about the virus. It's driven by our own unregulated inflammatory response. The virus replicates like crazy, viral loads spike, and our immune system essentially panics. That hypercytokinesia, the massive overproduction of inflammatory cytokines, ends up trashing the patient's own lungs and organs, just as much as, if not more than, the virus itself.
Now check out this data, because it perfectly illustrates a wild discrepancy in clinical outcomes. Historically, in the Western Pacific region, we saw a catastrophic case fatality rate of 66.3%, with the global historical average hovering around 48%. I mean, those numbers are terrifying. But look at the recent U.S. dairy outbreak leading into 2025. The fatality rate observed so far is sitting way down at 1.4%. Why the massive drop? It largely comes down to the route of exposure. Getting splashed in the eye with milk is very different from deeply inhaling high viral loads from poultry dust. Plus, we might be seeing some partial cross-immunity from prior seasonal H5N1 infections, actively blunting the severity here in the U.S.
Okay, Section 4, Zoonotic Transmission Routes. Let's break down the actual spillover mechanics.
So, how are human spillovers actually occurring at the agricultural interface? As this joint global assessment from the FAO, WHO, and WO shows, the sheer volume of virus out there in animal reservoirs is creating a persistent, literally unavoidable risk of multimodal transmission. We aren't just looking at wild birds anymore. The virus is pushed into terrestrial carnivores, marine mammals, and most critically, domesticated livestock. It's created this incredibly contaminated human-animal environment interface. To put that into perspective, let's break down the exact mechanical transmission pathway recently documented on California dairy farms. The virus basically found a brand new niche, end-linked sialic acid receptors located directly inside cow udders. Because of this, raw milk is getting pumped full of massive viral loads. Then, during active milking, those industrial machines agitate the milk and generate aerosolized particles right there in the parlor air. This turns into a severe occupational hazard. Workers are facing direct inhalation risks or taking raw milk splashes straight to the conjunctiva. We're even finding infectious virus persisting in treated farm wastewater. It is everywhere in that environment.
Finally, let's hit Section 5, Countermeasures and Vaccine Development. Time to look at our medical preparedness.
Right now, our first-line pharmacotherapy tools remain antivirals, specifically neuraminidase inhibitors like oseltamivir and PA endonuclease inhibitors like baloxavir. But the virus is always evolving, right? So virologists are constantly monitoring for specific resistance markers. They're scanning for the NA H275Y mutation, which ruins oseltamivir, and the PA I38M/T mutations, which tank baloxavir. What's arguably even more concerning is the sporadic emergence of polymerase basic 2 mutations, specifically PB2D701N and E627K, showing up in dairy cattle and wild mammals globally. These markers are huge red flags. They represent active, highly concerning steps toward mammalian adaptation. They basically tweak the virus's replication machinery so it can function much better at our slightly lower mammalian body temperatures. But there is genuinely good news on this front. Global preparedness is advancing rapidly. The US CDC has already synthesized candidate vaccine viruses, or CVVs, that are virtually identical to the hemagglutinin proteins of current circulating Clade 2.3.4.4b strains. Tests show these CVVs offer robust antigenic coverage and neutralization against recent human isolates. And as I mentioned earlier, our population-level pre-existing antibodies from seasonal H5N1 might actually be offering some partial cross-protective severity reduction, which is a nice bonus. Plus, we are actively leveraging cutting-edge tech. Moderna is already deep into clinical trials for their investigational mRNA 1018 H5 bird flu vaccine.
Which brings us to the ultimate question. We've mapped its genetics, we're tracking the transmission, but Clade 2.3.4.4b is completely relentless in its expansion across mammalian species. Will our proactive vaccine developments, like the mRNA 1018 H5 trials and our viral surveillance, outpace this virus's inevitable molecular adaptation to human alpha 2.6 receptors? It is the defining scientific race of our time. Thank you so much for joining me to explore the intense biology behind this evolving threat.